because sediment's made of a lot of different kinds of things. So you've got inorganic particles like clay or mud here, or silts. And then you have organic material. And so when marine snow lands on the seafloor, it can carry pigments with it that are greenish, darker, or lighter. And so when you see that mottled color, it's a sign of marine snow that's landed on the seafloor. And that's the tasty stuff for animals to eat.
It's all my fault.
It's all about perspective, because I look at that as that's stuff for animals to eat. And a geologist would look at that as obscuring the very interesting minerals and rocks behind it. So I remember when I took my geological oceanography class, and we were on a field trip, and there was a squirrel and some bushes that were there. And our instructor was like, this biogenic stuff. Take a look at the rock here.
Sometimes called bioclutter.
Bioclutter.
When I'm doing shipwreck surveys, archaeologists are always complaining about the bioclutter.
Is that the sponges?
Yes.
Oh, goodness. Bioclutter.
They're often needing to see markings on the ship's serial numbers, or just to identify a certain part of the ship.
Ah.
And they are upset that there's organisms on there.
Well, it's a hard surface.
Yeah.
Where there's a hard surface in the ocean, you're going to have something trying to grow on it.
Sanctuaries not only protect the marine organisms, but also the maritime cultural heritage, including shipwrecks and submerged airplanes. Our most famous one in our sanctuary is the USS Macon. It was an airship built by the Navy during wartime, or right between World War I and World War II. And the Macon was built, I think, in 1932, and unfortunately crashed off of Point Sur in 1934. And the airship remains are still there, as well as 4 of their Sparrowhawk biplanes. And we've been able to identify 3 of the 4 because their canvas on their wings are still there, and they were each uniquely marked. Yeah. Also on the seafloor, we see the ship's galley, the captain's quarters, desk, file cabinets, and many of the Maybach engines are still there, and the condensers that collected condensation and helped with the ballast.
So I actually hadn't made the connection until this year what an airship was. This was not a ship on the water.
Right, it looks like a blimp, but it was called an airship, or
So-
a rigid dirigible. A rigid airship or dirigible.
Oh.
So it's made up of aluminum frame covered by cotton covering that was used to transport these Sparrowhawk biplanes to help sleuth enemies at our shoreline.
So it was an aircraft carrier that was airborne,
Yes.
essentially. Holy cow.
There's another grenadier, likely Corphonortes armatus, the abyssal grenadier.
Not the rough one.
Well, it could be.
What?
The rough abyssal grenadier and the abyssal grenadier are hard to tell apart on video. The species are Corphonortes armatus and Corphonortes yakine. I'm going to take a guess that those are armatus. And that grenadier, I think, is Leptolepis, Corphinoides leptolepis, which is the ghostly grenadier.
That's fitting for October. Like that. Sponge alert. There's a sponge. That looks like a Saccocalyx. It's got a stalk at the bottom and these little finger-like processes sticking out off the sides. All of these are provisional identifications because you really need to collect a sponge to be able to identify it. They can take on so many different shapes depending on the water and flow conditions around them. They can be tricksy.
We trust you.
Oh, I'm saying don't. Not for an identification from video. But I'll get back to you on the ones that we collected yesterday. I'm really curious about that yellow-green sponge that we collected. Once we brought it up, we saw that it had these long tracts of skeletal spicules, they're called, of skeleton winding around the tube of the sponge. So they looked almost like bands going around it. It's just a really neat sort of architecture. Oh, and this is something neat. Some sponges have a rigid fused skeleton that's secondarily kind of fused or cemented together. And there was a study done by some biologists in partnership with some engineers that looked at the load-bearing properties of the arrangement of spicules, and they found that they followed similar buttressing principles that we use in architecture. So it's pretty neat. Like they have buttresses.
Guys, I'm going to take this opportunity as the cliff comes away to come away a bit and do some quick tether management.
Okay.
Okay.
Thank you.
Can you describe the tether management?
Just taking a turn out of our tether there.
So it ended up with a turn in it from us wrapping around the ridge for the last couple of hours?
Yeah, that and a combination of me dragging Argus around, so it just happens to be what way I drag it. Sometimes we pull it to keep the shot there.
Mm-hmm. Is Argus able to turn from side to side?
What's that?
Is Argus able to turn from side to side?
Yeah, we can control the heading on Argus. It has 2 thrusters. You can control the yaw.
Can we zoom on that anemone just real fast as we're going by?
So Antonella is controlling the Z and yaw of Argus, and myself, by moving the ship, controls the XY of Argus.
Okay, coming right.
I think that is Bolosera, and maybe Bolosera kensmithi.
Oh, wow. Ken Smith-I, named after Ken Smith at the Monterey Bay Aquarium Research Institute, likely.
Yes. Says this species was described from a specimen trawled at Station M. Based on the description, we believe this in situ image that we have depicts the species. So Ken Smith has done a lot of work at Station M, which is a long-term monitoring station in the deep sea, sort of west of Point Conception, I think.
Yes, and this is the 30th year that it's been monitored. It's I think it's the longest monitored abyssal study site. So you know, we do have access to the deep sea, but it's hard to keep and maintain. And in 1989, Ken Smith with collaborators put together a program to put out several moorings and monitoring equipment at Station M, and revisited 1 to 3 times a year to turn around the instruments to collect what was deployed down there. And with that, they have this amazing time series. And from that, they've been able to see that while things look slow and gentle here, slow currents, slow moving animals, really there's a lot of dynamics that happen on even seasonal timescales or between years. So they were able to see the seasonality of deep sea animals and behaviors. And with more and more years, they're able to see different timescales and the sort of effects that happen on different timescales.
We found a cave.
It is a mini,
Did.
a mini cave.
Oh, they do exist.
Looks like a very robust grenadier.
It does. And lots of crinoids again up here.
I wish crinoids lived on land.
Yeah,
I think running through a field of them would be really cool.
they're a little crunchy to sort of roll around in them.
Oh no, just like when they fly.
Ah,
Flying crinoids?
Not locusts, but crinoids.
yeah, so Miles is referring to swimming behavior that crinoids have. So if they're disturbed, the ones that are not on stalks, like these, will lift off the bottom and they'll wave their arms in undulating motion, and they look like these beautiful, they have this just beautiful swimming motion to them. They move super slowly and look like they're floating. Well, I guess they are floating there in water. Here's another Bolosera, a sea anemone on the left.
Oh yeah, different lighter color.
Mm-hmm. Wow. You guys have any estimate of scale? Like how tall is this cliff that we're looking at?
Well, we're pinging something below us at 5 meters, but you can see on the Argus camera,
Oh, okay.
it goes on below us, and I'll pop up to the top. Started at 12:53, or sorry, 32:53.
Mm-hmm.
I'm gonna run out of leash before I get there, though.
Wow.
You're starting to see it in sonar. There you go.
Got it. Oh, with the bathy-pathies at the top. And our depth now is...
It's been running around 15 meters, maybe. So.
Wow.
That was a nice
15
orientation.
meters tall.
Thank
Yeah.
you. It's good to see what's up top every once in a while.
It's hard to get a sense of scale from the close-up view, so it's nice in that way.
So we're paying for that a little bit. I need to catch up.
Okay. No worries.
That was great. Thank you. Ooh.
Oh, hi, moose octopus.
Moose.
See if I can get to the north of you a little bit there.
This is a great view.
Saccocalyx. Sponge alert. Well, I'm off to a good start for this next year of life. Seeing lots of sponges. Oh, you can really see the striations in the sediment here.
Dan, some people are commenting online about the gum chewing.
Sorry, sorry. Closed mouth, chew gum.
Miles, what does it mean on the sonar for Argus? Those scattered yellow pings?
It's just this noise. It's sort of the edge of how far we can see.
Ah, okay.
I mean, it's beyond 60 meters out, which is still pretty good. But
Oh, I see. The scale's different there. Whoa.
yeah, if you look in the upper right of each one, it tells you the scale of each division.
20 meters for Argus and 5 meters.
Yeah, so that
Yeah.
inner ring on Argus is 20 meters away.
Got
and
it.
the inner ring on Herc is 5 meters away.
There's it, Bathy Pathies.
Yeah.
There at the top. And another one.
Sorry about the speed here, guys.
No, I like it. This is
This is good
great.
speed.
Oh, really?
This is,
Uh-huh.
yeah, this is great. We get a good orientation of the features and
Yeah, it's kind of rough. We're just trying to get to the north of Argus. I've been behind for a long time now, and it's
no, this is great. I like this part.
roller coaster ride.
It's almost where it's as if we're in an IMAX theater. Flying through.
So we've got a Lepidisis coral
60-meter speed run there while we get back to the north of Argus.
and Bathy Pathies, the feather-like coral, the central stalk, and then branches coming off either side. We've got a bunch of those Bolacera sea anemones. Here's an octopus up on the top of the ridge between the lasers now to the left of the lasers.
Mm-hmm. Let's see if we can
Rapid zoom.
sex them.
Oh, it's curling up that arm now that we've stopped. Come on.
That's
Male.
male.
Him.
Male.
Or is it a damaged arm? Oh,
Looks a little damaged.
it does look damaged.
Not so sure. That's male.
Thanks
The
for
behavior makes it think male, sort of
Mhm.
protecting that
Yeah,
arm. Okay,
coming
thank
wide.
you.
Full wide.
I wonder if that could be a behavioral thing. Male-male competition. Try to destroy or damage the hectocotylus of your competitors.
Brutal.
Yeah. It's nature. Well, or if they could get damaged also from females from the act of mating.
Looks like we're going off the cliff. There's a nice black coral, Bathypathes, and some sea stars sporadically placed. Most of our previous dives, Davidson, have been at the top of the seamount. We haven't spent as much time down on these flanks and deeper portions of the seamount, so this is good to characterize what is here.
I did not see any sweeper tentacles on that Bathypathes.
Oh, thanks for looking.
My new favorite target thing to look for. This likely just wasn't known by me, but I was not familiar with Bathypathes having sweeper tentacles, which are these tentacles at the base of the stalk that are predator deterrents. So they sort of have extra defensive stinging cells down there that help keep predators from climbing up the stalk to get to the tasty zooids at the rest of the colony. So the temperature down here is 1.7 degrees Celsius. That's super cold, at least to me. So that's pretty close to freezing. But water doesn't freeze down in the deep. And part of that's related to pressure and part of that's related to salinity. So because it's salt water, it has a lower freezing point than fresh water would have.
And density.
And density, which is related to temperature and salinity. Yeah,
Right. Isn't water is most dense at 3 degrees? 2 degrees?
You mean fresh water? Or, because salt water is most dense at like negative 1.7, I believe.
Interesting.
Yes. So areas of the world where you have downwelling, like in the North Atlantic or in the Antarctic, water will get below freezing and become denser and then sink. Unless it freezes, in which case it's less dense and it floats. So that cold temperature relates to the activity level of the animals that are here too. So invertebrates, and I'm going to guess the fish here as well, because there aren't many fishes that do regulate body temperature.
Yeah, these don't.
Okay, so yeah, the temperature around them affects their metabolic rate. They're called poikilotherms. So if it's colder, then the metabolic rate is slower for these animals than if the water were warmer. So the animals that live in the deep sea tend to have slow metabolisms, which is reflective of the cold water that they're in.
And with fish down in the deep sea, they not only grow slower, but they also live longer than their shallow relatives. We've done some age and growth studies of the grenadier, not the deep species we see here, but the shallower species, the Pacific grenadier and the giant grenadier. And if I remember correctly, we were able to validate some of their ages up to between 30 and 50 years old, but presumably they live a lot longer than that.
Dang.
So when you're eating your grenadier from the supermarket, you might be eating your grandparent or your parent.
Someone who was born around the same time, hopefully. Not your actual grandparent.
Of
I'm
course
sorry.
not. The age
I'm
at which they are.
Yes.
Is there a market for grenadier?
There is. Pacific grenadier are often in the supermarkets when other fisheries are not, they're not able to catch other fish, like the sablefish fishery. If they can't get that at a certain time, they'll switch to Pacific grenadier. And they're sold up and down our coast from Washington to California.
That's why they got the name grenadier, because rat tail just didn't sound palatable.
In the market.
I've been hoping to see the ceryanthid tube anemones. And so I get tricked by the dark anemones, but whenever we've zoomed in on them, they don't have those two different sizes of tentacles sticking out. But they're in the field guide. So they're
Yes.
here. And we collected
Unless
a
we
piece
have
last time.
Did we?
We did.
Oh, I wasn't with you.
Right, yes.
Oh, hello explorers, I am back and I am so happy to see all of these questions that you guys have been sending in. Oh my goodness.
Welcome back, Danny.
Thank you.
You want to say where you were, what you were getting to do?
Yes, I had a awesome ship-to-shore interaction. So I pretty much Google Hangout into a school in Rhode Island, in Warwick, Rhode Island. It was the Community College of Rhode Island. Actually very close to where I went to college. I went to college in Bristol, Rhode Island. And they were, I think, awake and excited to hang out with us. They had some fantastic questions. And I see they're even writing in more now. So awesome. Welcome. Thank you for joining. What have I missed, you guys? Moose, octopus.
We covered
Yeah.
some of those questions.
We covered some of them.
The older ones.
Gotcha.
We explained the sonar.
Did you get the happy birthday, Amanda?
I did from
Happy
her
birthday,
from her kids at
Amanda.
MLML.
How deep can the remote vehicles go? About 4.
We did that.
We did
when-
that. Did you?
Yeah, 4,000.
Hello, Colorado.
That's about it. The rest
Crabs.
is new.
Okay.
We didn't do the crabs.
Have we seen any crabs? Yes.
Actually,
haven't
there's
seen
one in
crabs.
frame right now. It's a squat lobster, but I think it's a crab.
It is a
You're
crab.
right.
Yes. We're us and our common names. We're always a little bit off. We just say what it looks like, and then we learn something different later on. This is a squat lobster. Is actually a crab. And I'm gonna let the scientists continue from there because that's all I know.
Mm-hmm.
We are looking things up.
Hastily looking up the taxonomy of this is
That sounds great.
Phenomenal.
It has taken us about 3 hours to get down to this depth, and then we have been traveling along this ridge of Davidson Seamount for about 18 hours now, I believe, roughly. And so we keep looking. We are exploring an area that has never been seen before, so we didn't really know what we would find down here. So you guys are seeing it for the first time, as are we.
That is a long tentacle armed
Ooh.
Bolacera
It is.
anemone.
It looks
Oh,
like
wow.
mermaid
and
hair or
there's
something.
3 benthic siphonophores. I think someone, a couple, like couple watches ago called them sea dandelions. Little orange
Yes.
circles there suspended by filaments, kind of in the crevices below the at the base of this. What would we call this? Feature,
Call it a
furrow.
fissure.
Everything that starts with F.
Eroded
Crevice.
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 Crevices.
Ah, yeah, you can see
Crane.
2 more above the lasers and just slightly wider.
Hiding in there, yeah.
Yeah.
We see you.
Interesting. That must be the type of habitat they're in.
Mm-hmm.
Yeah, so squat lobsters are crabs within superorder Eucarida, order Decapoda, same as crab. Most crabs, or true crabs. Sorry, same as decapod crustaceans. And then they're within infraorder Anomera, which includes them, the squat lobsters, but also lithodid crabs and hermit crabs, the pagurids.
Decapoda being 10 feet?
Yes, that's right. So if you were to count the number of limbs, they would have 10 legs.
10 legs.
5 on each side.
That's a lot of legs.
There's a
I
lot of
guess that
legs.
includes the claws as legs.
Yes.
The big pinchers?
Yeah.
Yes.
It doesn't count sort of the mouth parts, right? Those are
No, those are separate.
separate. Okay. They've got some cool, and I'm blanking on the word for it, but very intricate mouth parts that they use to eat that are very interesting.
Count the legs.
1, 2, 3, 4.
There might be 2 in the
Might be
front,
reduced.
hiding.
Oh yeah, I think there are actually. You're right.
But yeah, tucked under.
Okay, coming like
So we're seeing some sort of pits and pockets along this wall. This is sort of a clay type sediment.
for what?
Are these pits a result of organisms feeding on the organic component? Once eaten and excreted, stuff may break the organic links and cause things to fall.
Well, we had a discussion about that earlier. We think maybe the clay is just falling. We don't necessarily think they're the same shape but different sizes, so perhaps it's just the clay falling
Sort of
off.
the structure of the clay breaking down. Oh, there's another sea star. Little squishy cuddly guys. Voracious predators, I know.
Yes. Just like sea otters.
Yes.
Cuddly looking, voracious predators.
But voracious predators. Yeah, I don't want a sea otter. They're so cute, but they are very territorial. What do deep sea flora and fauna do to kind of guard themselves against worse conditions? I don't know.
Well, part of it is having a slow metabolic rate. So, again, it's cold, and that helps keep their metabolism, well, causes their metabolism to be low. So they don't need to eat as much food as a more active invertebrate of the same type or the same species in warmer water, like near the surface. And the other thing is a lot of these are adapted to live in this boom and bust life area in terms of food. So a lot of the fish down in deep water, don't they have higher proportions of fat or
Lipids.
lipids?
Yep, for stored energy, yeah, they do. Especially the grenadier.
Well, and as harsh as we view the deep sea, it's actually a really stable environment.
Yeah, and they are adapted to it. So for them, it's not as harsh as it would be for us.
For us. We have a question from Kim from Norway. How do you pronounce the Latin word for flying fish?
Oh, okay.
Looks like the family name. Exocotidae? Exocotidae?
And we want the scientific name pronounced?
Yes. Exocoetidae. I am not great at pronouncing my biology words. I kind of make them up as I go.
Exocetidae?
I would say Exocetidae.
Okay, Exocetidae it is. So there
Kind
you
of-
go, Kim.
Yeah.
From Norway, she wants to name her sailboat Exocetidae.
Ooh, very neat.
I think that is a great
The
name.
flying fish.
Ooh, look at that fish. That is
Ooh.
perhaps another cuskeel. This is maybe the other species, or is it a snake? Or is it a?
It almost looks like he's using his pectoral fins as like...
Might be a zoarcidae or a snailfish.
Wow.
That's okay.
We hope the octopus garden will be long-term research too, Internet. Absolutely.
Oh, definitely. We know so much more about it having come here again, but there's still so much to learn about it.
Yes. Ugh. We would just love to stay here forever. This is so fascinating. So many new things that we can learn. We discover new species all the time, but it always takes us a little bit to understand that they are new species. So we may see something that looks similar, and then we bring it back or sample it or take the picture to an expert, and then we learn that it is slightly different than things we already know about. So we do get a chance to find new things, but we have to research our new things thoroughly before we confirm that they are new.
I think that fish was an eel pout. I think it was the snub-nosed eel pout, Pachycara bulbiceps.
Bulbiceps.
It's
I wish I
Sounds name.
had a better look, but that's my first guess.
Pokemon, that is a cool name.
Taylor Ann, do you have a frame grab of it we could take a look at? Okay. We can, we'll be able to look at the frame grabs after the dive, she says. So we can figure out who it is then. Something I wanted to add to what Danny was saying is that there was an estimate in one paper I read that probably only about 20% of species in the deep sea have names. And when I've mentioned this to other deep sea researchers, they've shrugged and said they estimate that it would be even fewer that actually have been described by science. It really is a frontier that is still being explored and is one of the least explored habitats, if not the least explored habitat on Earth, which is interesting because it's the largest ecosystem on Earth. And so, yeah, a lot of the time when we're looking at these things, we're giving them the closest name that we can think to give them, but that's how we keep on finding that once we take a closer look, this is in fact a new species, and that's in fact a new species. And I want to put it out to you guys who are watching this right now that if you see something interesting in our feeds, or if you are out tide pooling or scuba diving, you might also be looking at something that is an undescribed species. And it might be familiar to you, but isn't necessarily named or described yet. So I was scuba diving as part of a class in 2006, and we came upon this little critter living on a piece of red algae that turned out to be a cnidarian called a Staromedusa. And there are only about 50 species of Staromedusae described. I don't know if that's still current. I'm going to pull that one back. I feel like there's been a lot of work done on them since then. But we took it back and we looked it up, and it turned out to be a new species. And, you know, it was something that we had seen, and talking to other scuba divers in the area, they had seen them, and we just didn't know that it didn't have a name yet. So it's pretty cool. You guys should always explore and be open-minded about what you're seeing, and maybe think about whether you want to share it, photos of it with scientists, and see if it could be new.
That's really cool.
Yeah, we can discover new things. We got to describe it and name it. That was a really neat process.
Ooh, that's
What name did you give it?
That one, it's called Haliclistus californiensis. So it was found by Gilbert William in his PhD thesis, and he named it in his thesis, but did not formally describe it in a publication because he didn't have enough specimens. So it had that name already from the thesis, so we kept that name.
Okay.
That's great that you closed the loop.
Yeah, and actually it was really it was really great. We were able to get in touch with Dr. William, who was retired by that time, and we got to share photos with him and got his opinion on it. We invited him to be involved, but he was happy to have us describe it. And so, yeah, we got to close the loop on his PhD thesis. He passed away a couple of years ago, but his children wrote and said that he was really pleased that we had done that. We were able to complete that description.
That's sweet. That's very cool.
And there is a star medusa species called Manania gwilliami named after him. So I think that's really nice too.
I'm getting a few more questions in here from our community college in Rhode Island. I told them about our 2 new ROVs, Atalanta and Little Herc, and they will be a very similar setup to what we currently have with Herc and Argus.
Octopus down in
We
that
have yet
burrow.
to see any brine
That's right.
pools that I know of in this
This
journey.
guy's tucked away.
Yeah.
It would be very, very cool to see some. But I'm not sure if other expeditions have found any.
We may not
Okay
see
for
the
Zoom?
arm.
Is it an octopus in a crevice?
There is an octopus in that crevice, yes.
What a good spot to hide.
Might be moose octopus.
But yeah, it does look like the other moose octopus we were seeing.
All right.
Cool, thank you.
Thank you for the zoom.
Okay. Yeah, we're not going to be able to see his arm.
No.
Her arm.
No.
It's arm.
Real tucked up.
Very cozy.
Why is the ocean important to you? I'm gonna let you guys answer this. What do you think? Why is the ocean important to you?
Wow, there's so many reasons.
I agree.
I'm gonna pick one. The first one that comes to mind, it's important to me because it's really, really important to all of us being able to survive. It helps regulate the climate that's here on the planet. It's responsible for half of the amount of oxygen that's produced each year. So from phytoplankton blooms and algae. So it's just,
It's probably the
Gosh.
most important reason it's important.
yeah, okay, that's true. But there's also a lot of intrinsic value to it that's,
Of course.
you know,
That's our primary function.
yeah,
Keeping the planet clean and
right.
air clean and providing oxygen.
But it's also beautiful. It's an amazing frontier that is alien for us humans. Is that another octopus?
Oh, octopus down the lower right.
And so, I mean, I think that it's just, it's a really cool, in addition to being really important, I think it just has value in being really cool, interesting habitat that shows us, it opens my mind to what's possible in terms of what life can be like and how things can exist. Anyone else want to weigh in?
I think those are great answers. I just find it inspiring. I think there are so many animals that have adapted to different lifestyles. Deep sea, pressure, cold, incredibly warm and sunny. They inspire me for a lot of my creative side of things, of how can we problem solve, because there have been so many creatures that have made challenges like that work, and I love to learn about the different adaptations.
I lived on the coast my whole life, and I just feel like I get a fresher air when I'm near the coast. I haven't lived in a landlocked state, but I've visited many, and I always crave to get back to the coast, to be near the ocean. Of course, it provides a place for me to work, but probably because I'm most interested in being near the ocean and studying the ocean. That's why I got a job in the sciences, the ocean sciences.
I do have a lot of recreational activities that are on the water. I like to scuba dive, that one especially. but I did live in, I moved to Edmonton, Alberta for my PhD, which is in a landlocked province in Canada. So we were about a 14-hour drive to the ocean. And so it is wonderful to live nearby and be able to have this access. But I also want to reach out and say people who live in the Midwest, like Taylor Ann, or people in Edmonton, Alberta, also get a lot of benefits from the ocean and can study marine science there as well, if this is the kind of thing that you find really interesting. So for me, living in Alberta, we would get ship cruises and we'd go out to the coast for our ship cruises and then bring our samples back to analyze at the university. And we also were able to go out to field stations to be able to study animals in shallower waters.
And I'll add to that, just because you're not by the ocean, this is your sanctuary. This is the national treasure. It's there for future generations, so hopefully at one point in time you'll get to visit a sanctuary closest to your home or from far away. I haven't been out to the Hawaiian Islands or Papahana, I can't even say the name, Papahana Hukuhuhumea, something. You're good.
Yeah. Sounds good. There's a lot of
I would love to get
syllables.
out there. Yes. Oh, great. Yeah, I think that's the snub-nosed eelpout, Pachycara bulbiceps. Thanks, Miles.
Yeah.
Oh, they were able to bring up the frame grab for you.
Yeah, it
great.
takes about 15 minutes for the server to sync, but we can look at those here.
Very
You guys
neat.
could look at them back there if you want. I can direct you.
Ooh.
That'll be the provisional identification.
Pachycara. Pachy is rough. Do we know what cara is?
I'm starting to
Head.
second guess my my ID, but
Rough head or rough face.
that's my guess.
So we're getting a lot of questions about the pressure that we are dealing with. So it is an increase of 1 ATM, 1 atmospheric pressure every, what did we say, 30?
10 meters,
10
30
meters.
10 meters, 30 feet. And we are about
Ooh, giant picniconid. Sea spider.
little over 3,000 meters down. So I'm gonna let you guys do the math. And that's the pressure that we're dealing with. Who is doing most of the talking? Well, I think we're all talking.
Amanda's just
Ah.
University of Amanda Khan.
Yes.
I want to go to your
I
college.
just jabber. But Danny's been doing a great job. I think it's you, Danny.
Thank you. Well, Amanda is teaching us so much about invertebrates. I can't tell you how much I've already learned on this trip. And then this voice that you are hearing is Danny Bailey. I am your Science Communication Fellow.
It's a coral.
Or one of your Science Communication Fellows for this voyage. If some of you are awake before, you
Should we
might
take a
have
look at
heard
that
my
Yeah,
friend and
we
wonderful coworker,
Can we take a
Scott
look at that
Simon.
coral?
And in a short while here, you will also hear Megan Cook,
Yep,
and
sure.
she has been a fantastic mentor to me. If you ever need someone to mentor you, oh my goodness, she is my favorite. There are so many lovely ladies that have helped me out. I don't want to forget them, Sam and Kelly and Allison, but it's been
That
such an
might be the
exciting adventure.
Chrysogorgia pinnata.
Ooh, that's really pretty looking.
Yeah.
Is that the
I think that's a Chrysogorgia.
the one that kind of looks like Christmas lights?
Yes.
Okay. That's how I remembered it. Chrysogorgia?
Chrysogorgia.
Chrysogorgia.
And it's in one plane. I think that's pinnata.
Oh, it is pretty.
All right. Yeah, that's what that is.
I wonder who lives in that hole to
Ooh,
the
there's
right.
benthic ctenophores on the left of the screen. You can see them.
Yes.
They're tentilla sticking out. Tentacles with tentilla. And, oh yeah, hello benthic ctenophores.
Here we go. Going in.
Wow.
That's full zoom.
We got good zoom.
Yeah, it's very delicate with these regularly spaced polyps. All right, thanks for zoom.
Okay. Coming wide.
We'll keep
Where
moving
are we
along.
in my question?
Okay, full wide.
Are the unknown species effectively subspecies or types of existing species that would be totally unique discovered types?
I think it varies.
Yes. So some
Yeah.
of them are subspecies. They're related to things that we already know. Some of them are their own unique new species, but do have similarities. What's that thing in the back by the squat lobster?
That would be a rock.
Oh.
I like rocks.
Well, all right. Rocks are pretty cool. They're very important down here for substrate, absolutely. But I thought it was something like coiled.
Ah,
I still want to see a hagfish,
Yeah, I think that's a rock.
but that's a rock. All right, go rocks. Oh, I think this is a great question. This is a question for everybody. So several years ago, there was that tanker that dumped, I don't know how many thousands of rubber duckies into the ocean. Do you guys remember that?
Yes.
Has anyone ever seen one of the rubber duckies?
of us individually?
Yeah,
Oh,
I have not.
I have not either.
I haven't either.
No. Has the person writing in seen one?
Oh, I don't know.
The
Person
famous
writing
rubber
in, if you've seen one, we want to hear the story.
So that was the result of a lost shipping container. We actually had about 15 shipping containers fall within our sanctuary.
Oh, no.
And one we've been studying. Mbari found one of them. We haven't found all of them. Just outside the Monterey Bay Sanctuary. And it is full of tires. And we are not opening it because that would probably cause more disturbance. And we're not bringing it back up because that would cause, they'd probably fall apart upon retrieval. But we are studying the long-term effects of that structure in an otherwise soft bottom community.
Oh, an octopus.
And we're also
Octopus.
studying the
Yes.
decomposition of it.
Can we zoom on the
Yeah,
arms?
can we zoom on that octopus?
Well, that's a great way to make
Yeah, let me get
the
closer.
best of a situation. Hello, octopus. This one's all curled up too. So we went from seeing a bunch that were sort of like splayed out and spreading out their legs to a few that are all curled up and tucked in.
Yeah, we may not see that third arm.
Where am I in relation to Argus? I'm south of it, right? I'm pulling you, am I? Yeah, we're not going to be able to get a steady shot.
That's
there
fine,
'cause
that's fine.
You can
Okay.
zoom a little bit, but
All right,
I'm
let's see what
working
we
on
can do.
Argus.
It's
We'll
okay.
make the most of it.
That's about it there.
I don't think we have a good
Hold what you got
viewpoint
there.
of that anyway.
Yeah, it's the wrong side.
That's
to-
okay. You can
to-
zoom out. Thank you.
Carry
Okay,
on.
coming wide. All right.
Am I under you or above you? Or, oh, you're looking we're totally stretched out. All right.
The rubber duck person said no, they live in Central Texas, but they saw the episode, or saw information about it on an episode of a
the
TV
ship's
show.
moving
Ah.
to the north, that's what.
It would be very neat to see one of those famous rubber ducks.
It would be very interesting. I would love to have one of those.
Oh,
Take it out of
that's
the
my
ocean, of
bad.
course. Oh, my rock friend. Where would a rock like that have come from? Seems like it would be a rock of probably volcanic origin from this Davidson seamount that had broke off in some point and been rolled down the slope or been cleared of debris
Kick off
for
your
some
auto heading
reason
until I
and
get some slack in the tether.
hanging out there.
Thank you.
It could be related to a glacial erratics, which are things that were dropped by glaciers long ago, made their way down rivers and into the ocean. They could be submarine landslides
What's
or things that gave way. So here we're at the skirt of Davidson Seamount, so it's possible some part of that broke off, tumbled
Yeah.
down.
Well, probably wasn't.
Lots of possibilities.
If you look at it when you drain them down, it goes down to 2, and it moves about that much from. It's very non-linear. Come down 5 minutes real quick for me.
I have a question about sort of an ROV disturbing or kicking up sediment. So has the ROV following an ROV, so Argus following Hercules, shown an increase in activity in clouds behind it? Well, I know we are like brushing up sediment occasionally as we go. Not all the time, but I don't know if anyone's
keep
studied
coming down.
that.
It's possible, but I would say
Yeah,
this is
keep coming
probably the lowest impact way to survey the deep sea.
Yes,
Other methods would be
absolutely.
things like research trawls, which is a net
Keep
dragged
coming down.
across the bottom to be able to pull up what you see. Things get torn up, you know, denude the seafloor, like it's going to have a much greater effect. But can sediment get kicked up by the props? Yes.
And our most current question
Yeah, that's
is,
good.
how would you know if the rubber duck is from the shipping
Hold what you got
container?
there.
Ooh, I don't know that.
There's lots of different pictures. For me, they didn't look like they look like stereotypical rubber ducks, but they looked a little bit different. I guess if you were in the same sort of area,
There might be lot numbers as well.
there may be lot numbers. They might be imprinted with a number that could tell. At this point, they would probably be in rough shape and pretty sun damaged if they're still out and about.
How long ago did that happen?
I do not remember.
27 years ago.
Really?
Mm-hmm.